Hello, this is Adam Sigler, and it's March 12th. It's 6 days after I presented my dissertation work in my defense presentation, and I'm going to capture it here while it's still fresh. And I'm excited to share with you what I've been doing for the last 8.5 years, and specifically talking about water and nitrogen movement through this non-irrigated agricultural ecosystem in central Montana. I want to start by recognizing my PhD committee, beginning with my advisor, Dr. Stephanie Ewing, who did an introduction that day, and then Clayne Jones, Jack Brookshire, Rob Payne, who were in the room, and then Gary Wiseman, who was on the phone from University of New Mexico. I also want to thank the funding sources that made this work possible. And so all forms of life on Earth require reactive nitrogen. And while right now we are all breathing 78% nitrogen, that is largely unavailable because it's all dinitrogen gas, tightly triple bonded. And so it's effectively inert and not available to most organisms except those with the nitrogenase enzyme, which evolved over 3 billion years ago and likely made it possible for more complex life forms to evolve on the planet. Those organisms we might think of as being associated with legumes, and that's the primary way that reactive nitrogen came into our ecosystems until the early 1900s, when the Haber-Bosch process came on the scene, where we placed the atmosphere under high pressure and temperature and fixed dinitrogen gas into ammonium, effectively adding a new human component to the global nitrogen cycle, where now with the growing world population, we have over half of the world's population eating food grown on Haber-Bosch nitrogen. A similar increasing pattern in fertilizer application in the United States, and an associated increase in groundwater nitrate concentration, indicating that as we add more reactive nitrogen to our soils, they become more leaky and more nitrogen gets lost to groundwater, and in many systems that also moves on out to surface water. This has been cast as a wicked problem, which has been defined as one where competing goals are at play within complex systems where there is information uncertainty. And so these two competing goals might be cast as protection of the water quality that we rely on while feeding the population. And so we often see plots like this where on the x-axis we have increasing nitrogen fertilizer application, and on the y we have increased crop yields, and we see this positive correlation, which is why we apply this fertilizer to feed our growing population. However, at the same time, that means we're getting increasing losses to groundwater, which is present across agricultural watersheds in the United States, where 20% of shallow wells in agricultural areas are above the drinking water standard of 10 milligrams per liter. And this is a trend that has been increasing over time. If we think about surface water and think globally, all of these little dots are places of hypoxia or eutrophication in our marine systems fed by loss out of surface waters. And so this is a widespread issue where protection of water quality seems at odds with growing enough food to feed a global population. However, when you talk to farmers, you can learn that you can learn a lot, for instance, in my case, by talking to farmers. You can learn that they're really good at logistics. You can learn that farming's complicated. And you can learn that they have a goal of getting all of the nitrogen possible into the crop. And that means minimizing losses to all pathways, including losses to water. And so if you have this conversation, you may begin to realize that there's a shared goal of nitrogen use efficiency. And so farmers are able to they know a lot about these systems and a lot about the limitations to improving nitrogen use efficiency. And scientists, when they become involved in the conversation, know about the broader context of the system and can bring the scientific method to bear on addressing these questions. And so at the Land Grant University, we are uniquely poised to address these issues, where we have students who engage with researchers who are connected to the communities through outreach and in the context of extension often. So we're able to bring these groups together to address these wicked problems. And as a water quality specialist, I often think about Montana in terms of our vast surface water resources. But I also think about the state in terms of our vast groundwater resources, where in many of our counties, an important fraction of the population receives drinking water from private wells. For this reason, one of my largest programming areas is private well owner education, or the Well Educated program, where this was one of the earliest programs that I picked up in 2005 based on previous work that my predecessor Jim Botter had done, where we guide well owners through testing and understanding their water quality. And we've reached over 7,000 participants in that time. And while the primary goal of this programming is to educate well owners about their water quality, we also end up with this data repository where we can make plots like this one in 2011 that showed very high concentrations of nitrate in Judith Basin and Fergus counties in the center of the state, similar to results that my predecessor Jim Botter had found in the 80s. And so it was some of this well-educated data, among other data sources, that's represented on this map of the Judith River watershed, where these darkest blues represent nitrate in drinking water, in groundwater above the drinking water standard of 10. In 2011, when this work started, we also had this data set from the M1 well collected by the Montana Department of Agriculture, showing nitrate concentrations in 1994 at around 10 milligrams per liter at the drinking water standard, and those numbers doubled over the next around 20 years up to 20 milligrams per liter in 2011. So it was data like this, as well as relationships that myself and Clayne Jones, soil fertility specialist, had in the area that set the stage for a successful proposal to USDA, led by Stephanie Ewing, and then also joined by Doug Jackson Smith, a rural sociologist out of Utah State University, to address three questions. The first, to better understand these sources of nitrate in ground and surface water. Two, to evaluate farming management practices that could reduce nitrate leaching losses. And three, to do this in a participatory research approach and framework where we involve the local stakeholders and farmers in the research throughout the process. And so this work would not have been possible without the intensive cooperation with the local advisory committee made up of Dave Wichman, the former superintendent of the Central Ag Research Center, local county extension, local NRCS, a number of local producers, and also the Producer Research Advisory Group with 6 farmers that we met with many times per year, and all of the farm management research happened on their farms. And so this work would not have been possible without the incredible cooperation of these partners. And so I'm not going to focus a lot the rest of today on the participatory part of this work, but it's critical to say that this work wouldn't have been possible without these cooperators. And so the overarching dissertation question is where, when, and how much water and nitrogen are moving through the soils, groundwater, and streams of the Judith watershed, and what are the controls on these fluxes? Or put more simply, what are the opportunities for increased nitrogen use efficiency? So my dissertation outline begins with an introduction, which we've already talked through most of, and then there are 3 papers. The first of which was published in 2018, which works at the landscape scale to quantify the amount of nitrogen and water that are lost, to give us a big picture framework to understand and start with, to know where the losses are, how big the losses are. That sets the stage for the second paper, where we do intensive modeling work to understand the interaction of management, soils, and weather as controls on water and nitrogen loss from the landscape. And this paper is in the process of submission now. And then in the third paper, we look at the gas losses of nitrogen back to the atmosphere through denitrification, and what are the drivers of that across this landscape. And then we'll wrap up with some general conclusions. So the study area is in the northern Great Plains, which is the green area in this image, which most is in the headwaters of the Mississippi and the Missouri River basins. And then we're within the Judith River watershed here, which is in the very center of Montana. And in the middle of that, we have the Moccasin Terrace here, where at the center of this is the Central Ag Research Center near the town of Moccasin. And so we can think about this terrace in a more conceptualized sense as being perched on shale, where it's isolated from lateral input of groundwater, so that all of the water in the aquifers under this terrace are derived from precipitation infiltrating on the terrace surface which is primarily in cultivated land use. And so it's this beautifully isolated system where we can understand how annual cultivation interacts with soils to control the losses of water and the chemistry of that to groundwater and surface water. So these streams draining out of the terrace are fed exclusively by groundwater coming out of the terrace. And so to understand what's happening here, we need to understand, with water and nitrogen loss, we need to understand what's happening in the soil. And if we zoom in at the pore scale, there's always some water left in soil, even when evapotranspiration has pulled out as much as it can. But then precipitation comes in and it starts to wet up the soil. And once the soil reaches field capacity, water starts to drain out the bottom as deep percolation. So this is half of the story for getting nitrate leaching, is water moving down. The other half is to have nitrogen present in soil that is mobile, and that often happens in the form of nitrate. And so we were removing nitrogen in harvest for many decades in Montana before we started adding fertilizer. We were taking nitrogen off, however, in that crop, and that was all coming from mineralization of soil organic nitrogen. Beginning in the mid part of the 1900s and really ramping up in the 80s, we started to add more fertilizer. So that then is another source of nitrate in soils along with mineralization. Nitrate is very soluble, and so when there is deep percolation happening and nitrate is present in soils, then leaching will occur. And in this system, we're talking about that being basically water and solutes or nitrate that have moved below 1.5 meters or about 5 feet. And so this is not only what's leading to those challenges with nitrate in groundwater, but it's also a lost opportunity for crop productivity when that nitrogen is getting down below the root zone where the crop can get it. And so how much water and nitrate are lost have everything to do with the soil water holding capacity, which is directly related to the thickness of the fine textured clay loam soils. Because as soon as water and nitrogen get down below that fine texture into the gravel, it moves readily down to the shallow aquifers. And so the thickness of this fine texture is variable across the study area, but in general is quite thin. So in this work, we've made a number of observations across soils, groundwater, and surface water. And so in soils, we've made measurements within intensely studied fields managed by our cooperators, where we dug many soil pits and we described those and characterized the soils. We installed suction lysimeters, and then our cooperating farmers farmed over the top of those lysimeters. And these basically allow us to, there's a tube connected to a bottle on the surface that we can access and are able to extract water samples from deep in the soil and then analyze those for chemical solutions, nitrate, etc. And the important context for this is that they were either collected during a time when crop was present or during fallow, and they were either from relatively thin soil installations or lysimeters installed in relatively thick soils. So that's the soils piece. In groundwater, we have access to some shallow wells, and then we also have locations where the aquifer intercepts the edge of the terrace and a spring is formed where we're able to sample. So we have a total of 7 groundwater sampling sites across the terrace. Observations in streams, we measured discharge, and then we also collected samples at 2 locations on one stream on Louse Creek, and then at a single location on a second stream, Porter Creek. And so these observations allow us to ask the question in the first paper of how did the dynamics of water and nitrogen at work in soils manifest in the loss patterns of water and nitrogen at the landscape scale. And our approach for this is to measure discharge in streams and then the amount of flow and then the nitrate concentration in those streams to get losses from the landscape. However, we know that there can be important losses of nitrate in the riparian corridor along streams. So we're also going to look at the broader groundwater nitrate concentrations before loss in streams occur. And so in this figure, we have 3 years of data from the 2018 paper, where across the top we have precipitation. And the black line is the water level in the M1 well. And so you can see that it bumps up in the spring when we have high precipitation periods. So groundwater level is responding to precipitation on the terrace surface. So groundwater is being recharged by that precipitation. We then measured discharge in Louse Creek and Porter Creek, and we used simple hydrograph separation to identify this darker blue area at the bottom where we're confident that the stream flow is coming from groundwater. And we're able to then use that to determine that between 5 and 10 centimeters per year of the precipitation falling on the terrace deep percolates or makes it through the soil. That's of the 38 centimeters of mean annual precipitation. In other words, 15 to 25% of annual precipitation is recharging groundwater, moving through soils. So then we're able to combine that flow of water with the concentration of nitrate in both ground and surface water and multiply those together to get the losses of nitrate out of this landscape. And so we'll start with the measurements in stream, which are these dots down here, which are multiplying the flow in the groundwater flow by the stream concentrations. And that allows us to estimate that between 5 and 20 kilograms per hectare per year are being lost from this landscape in the streams. But we know that there can be losses in the riparian corridor, so we're going to use the concentration of nitrate in groundwater, so the mean here is the black line, and we're going to multiply that by flow, and that's going to give us the black line down here. That allows us to make an improved estimate of the losses from soils of 10 to 20 kilograms per hectare. So that's numbers corrected for riparian losses. And this is compared to fertilizer application rates around 90 kilograms per hectare. And so for those used to thinking in pounds per acre, the conversion factor is 1.1, so the numbers are very similar. And so these numbers at 20 to 30% of a possible fertilizer application rate are becoming agronomically important. And it's important to note that these are not just losses of fertilizer. There's an important soil organic nitrogen story that we will return to. But so thinking about those losses, if we focus in on this period where we have the highest confidence that stream flow is coming predominantly from groundwater, we can see that these dots, which are the in-stream nitrogen loads, are always below the black line, indicating that there is always a loss in the riparian corridor. And we can calculate that out to estimate that 50% of the nitrate that is coming into the riparian corridor from groundwater is being lost in the riparian corridor. And the nature of that loss we'll come back to later on. But the losses are much lower for Porter Creek at 15%. And so this work at the landscape scale gives us a really good idea of the overall picture. And it's interesting to know we have this loss in the riparian corridor. We also know that these losses in soils are much lower than losses estimated by Andrew John, Clayne Jones's master's student, which were much higher rates of loss, around 50. And so we'll come back to those unexplained differences in losses in a little bit. So this is what's happening as a big picture at the big landscape scale, but what's happening in where, when, and why are we getting these losses out of soil? And so this leads us to the second paper where we ask, how do management, weather, and soils interact to control these losses of water and nitrate? And so we're working on the Moccasin Terrace again. We're also working on Field C, which is a cooperator's field near the town of Moore. And so these three pieces, management, weather, and soils, let's start with management. A atypical rotation in the study area is spring grain, followed by fallow, followed by winter wheat, and then if it's a 3-year rotation, then that repeats. And we know from previous work, including that by Botter et al., 93, that it's that fallow year where we expect the largest nitrate leaching losses. And if we think about why that is, if we have here looking over time, the green area is when crop is present. And in a spring grain year, that crop is going to take up the water and nitrate. So when the crop is finished growing, the soils are relatively dry and there's relatively little nitrate. In the fallow year, we don't grow a crop, and we're talking here about chemical summer fallow. So all vegetation growth is suppressed for an entire year, over an entire summer, so it's even longer than 12-month period. And this is largely motivated by storage of water for the subsequent crop. The validity of that motivation we'll revisit later in the talk, but basically in this year we get most of our precipitation in May and June, and so the soil gets full of water, but it also gets full of nitrate. There's no fertilizer added, but the soils are moist and they get warm, and so the microbes are very happy and they get busy mineralizing soil organic nitrogen. So during the fallow year, we get the soils are full of water and nitrate are primed for leaching loss when we get subsequent precipitation. So it's this high risk period, and it carries over into the next year because often we get a lot of our spring precipitation before those winter wheat roots are fully established to take up that water. And then the cycle continues. And so we've basically got this rotation, which is fallow, winter wheat, spring grain, but we can think about this in simpler terms of fallow year, crop year, crop year. But I just said that what happens last year also is important. So I'm going to talk about these in 2-year sequences where I'll present results for a single year, which is the second word, the underlined word, but I'm also telling you what happened last year for context. So this is one way that we could see the rotation on the landscape. We could also have the exact same rotation with fallow offset by a year. So it's basically just the same year, the same rotation offset in 3 possible different time offsets, which you might see spread out across the landscape. So this is the management piece. For weather, we're basically talking about precipitation and potential evapotranspiration. So we get the majority of our precipitation in the study area in the months of May and June, and so that's the water coming into the soil. The water leaving is potential evapotranspiration. So that is wind speed, solar radiation, temperature, and relative humidity conspiring to determine how much water the atmosphere wants to pull back out of the soil and the plants. And this peaks in the warmest months in July. It's important to note that in a semi-arid system like this, potential evapotranspiration is anywhere from 2 to 10 times incoming precipitation. So a lot more potential to pull water out than what we have coming in. So that's the weather component. Moving on to soils, which we know something about at the landscape scale because of NRCS Surgo data, where we're going to focus in on the thickness of fine texture. And so thickness, that's length in the Z direction here of fines, so Z sub F. And that's the thickness of what is clay loam soil here. And up in this northern catchment, the Louse Creek catchment, we have these darker beiges, which are the relatively thicker Danvers series. And then in the southern catchment, Porter, we have more of the thinner soils in general. And we also know something about soils at the field scale, where on field C we have this greenness image from a NAPE image shot in 2011 when a barley crop was growing. And we hypothesized that the greenness is due to water availability related to how thick the fine textures are so that it can store water and make it available for the crop. And so we dug soil pits across this field in 2012, and we plotted the greenness against our observed soil thickness, and sure enough, we see a statistically significant relationship for the shallower soils, which allows us then to add in a depth aspect to this map, where down here we have a histogram of the depths where we can see the percentage of the field at different soil thicknesses related to those different greenness or NDVI indexes, normalized difference vegetation index. And this tells us that the section of the field that's shallower than 25 centimeters is 13%. There's 30% at this intermediate depth, and this fraction of the field with greater than 50 centimeters thickness of fines is 57%. This relationship in this map was validated by extensive additional soil sampling that we based on this map. So we have some confidence in it. We then move on to our modeling effort. We're going to use Hydrus 1D to model water movement through soil, where in simple terms we tell it how much precipitation is coming in on a daily basis, we tell it how much potential evapotranspiration the atmosphere is trying to pull out, we tell it some things about the hydrology, behavior of the soils, and it tells us what comes out the bottom as drainage, as well as what's happening with soil moisture on a daily basis. So we've also made extensive measurements with eddy covariance of actual evapotranspiration. We've also made measurements in soil from our instrumented soil pits of volumetric water content. And so we're able to compare those modeled numbers to our measured numbers and to see how the model's doing. And in simple terms here, I'll just say that the model does fairly well. And so we have these three rotation scenarios. We then have 15 years of precipitation and evapotranspiration data from the Agrimet station at the Central Ag Research Center. And the reason to have these three different rotations is it's so that each land use fallow, for instance, is going to fall on every single possible weather year. And then the model results will average all of those together. And we're going to vary soil thickness from 5 to 150 centimeters in 5 centimeter increments, so 30 total depth increments, giving us a total of 90 model scenarios, which are the results. I will be averaging those together to present you the results I'll show in a moment. The model is going to give us precipitation partitioning. In other words, what are the loss pathways for that precipitation that's coming in? And so one of those is transpiration, or water loss to the atmosphere through the crop. As a farmer, this is what I would want to be maximizing because transpiration is directly correlated to yield. But we also lose some water to the atmosphere by evaporation, or water lost directly from the soil surface, which imparts no benefit to the crop. And then we've also got the balance is primarily going to drainage. Again, that loss below 1.5 meters or 5 feet where the roots can't get it. There's very little runoff over the surface in most of these very well-drained soils in the study area. So let's think about how this looks for our spring grain year, or the crop following crop. Again, we have 38 centimeters of precipitation on average. And if we say for this scenario that we've got 25 centimeters of soil thickness, or about 10 inches of soil, our model results tell us that 7 centimeters of that 38 are lost to drainage, that 12 are lost to evapotranspiration, and that 19 are lost to transpiration. So we're going to take the same configuration, and now we're going to look at that for different values of soil thickness, that Z sub F value. And so here we have increasing soil thickness from just 5 centimeters over here to 150, or about 5 feet of thickness. And on our y-axis here, we have those loss pathways that we just talked about. And then this horizontal line is our mean annual precipitation at 38 centimeters. So we can see that for our thinnest soils, we're losing over 10 centimeters of water to drainage. But in our thicker soils, we're losing almost none. Evaporation, we're losing over 10 centimeters of water all the way across. It's fairly consistent across our soil thickness. And then transpiration losses for our thin soils is pretty low, and that's because those soils just can't hold on to water. It gets drained out the bottom and the crop can't use it before it's lost. As we increase in soil thickness, however, transpiration's going up, but overall, our losses for that crop year, or the spring grain year, are equal to inputs. Basically, the bar heights are pretty close to equal to the mean annual precipitation. If we look at the fallow year, we see a different story where deep percolation has jumped way up, losing over 15 centimeters from the thin soils and approaching 5 centimeters from those thicker soils. Evaporation has jumped way up and again is large across soil thicknesses. This white space below the line now is storage. We're not losing all of the water in those years. And so transpiration is absent because this is a fallow year, no crop or vegetation present. And so this storage of water is one of the motivations for fallow. But notice that for soils less than 25 centimeters, or about 10 inches, we're storing no water on average. So there's no water storage benefit for those thinnest soils. And even in the thicker soils, we're only storing less than about 10 centimeters of water. That means that we're losing 75% of the incoming precipitation in those fallow years. Not a good statistic if you're trying to use all the rain you can in a semi-arid environment. If we look though then at the winter wheat year, or the crop year following fallow, we see deep percolation or drainage and evaporation patterns relatively similar to that first year that we looked at. A few subtle differences but transpiration now bumps up. on the thicker soils. Thin soils, not so much, but on thicker soils, we are getting an increase in transpiration, which is basically the mirror of that stored water in the fallow year. So this is one of those reasons that we do see a bump in winter wheat yield following fallow as opposed to following crop. So the thing that we're primarily focused on today is deep percolation losses. So I'm going to take these lines for this dark gray deep percolation loss and place them all on the same plot where we can see now deep percolation on the y-axis and again soil thickness on the x. The largest losses overall are for these thinner soils over here. And then the other thing that we saw before is that these crosses are the fallow year, and so the deep water losses from soil are the highest in that fallow year across soil thicknesses. We can now take this and apply it to what that means at the field scale because we have this depth map for field C. And so if we want to think about what's happening here on average in the fallow years versus the crop years, let's take an example of 7.5 centimeters of deep percolation. And we come over here and hit the crop year, and that gives us a soil thickness of 19 centimeters, which is so the sections of the field that are thinner than 19 centimeters are just these very lightest colors here. And so in a crop year, that's the fraction of the field that's losing on average that 7.5 centimeters or more. Whereas in a fallow year, we're going to bump all the way over for 7.5 centimeters of loss to this point, drop down, that's 55 centimeters, or now 60% of the field. So that is all but the darkest greens here. The dark greens are the thickest soils, and in this fallow year, everything lighter than that is deep percolating 7.5 centimeters or more. So we're dramatically increasing the area of the field for any given loss rate of water. So this information, thinking about how rotation interacts with soils to determine how efficiently we're using our precipitation, starts to be pretty useful when we're starting to talk with cooperating farmers about water use efficiency. And so to summarize this, if we think about the field in terms of thin soils and thick soils and the different years, it's those fallow years on the thinnest soils where we're losing the most water. And in contrast, it's the thicker soils in the crop year following crop we're losing almost no water. The crop is using it much more efficiently. We're losing much less to drainage. So thus far, these results are thinking in averages over that 15-year period that we have data. But we can also think about this, how it plays out over time. And so if we take, for instance, a soil that's 55 centimeters thick, or about 22 inches, and we look at those results, over time, it's for the crop year following fallow or the winter wheat year, we can see where we have these big losses. Anybody around in 2011 remembers that was a very wet year, and we see this dramatic deep percolation loss, or this dark purple here. We also have some losses in 2013, 2014. We can see what that looks like for the spring grain crop, which is relatively similar, a few subtle differences that I won't get into just now, but where we see the biggest differences are in the fallow year, where we see losses in years that we really didn't see if there's a crop present. And so in this case, actually, if we had a field in a 3-year rotation, we get dramatic losses in 2011, and then 3 years later, it would have been in fallow again, and so that a field that happened to be in that rotation in this very wet year, 2011-2014, loses a lot of water. So the results of this modeling work is valuable for us to be able to think about when we made a lot of observations in our study, but you get the comment that was a really wet year, or that was a really dry year. So how do we know how that every year is weird for some reason? So how do we know how those study results what they mean? And I mean, our cooperating farmers would say if you could control the precipitation, then we could really nail this nitrogen use efficiency. And what a model lets us do is control the precipitation. We can look at all the different scenarios. And this is useful because if we have a trial study that happens to come out on the ground in 2012, which is a very dry year, or in 2011, a very wet year, or an average year, more like 2013, we can see how that how those results compare to what we might see in a more average or in an extreme year. So this really can allow us to see the benefit that a model and model results can show us. Okay, so this is the water loss story from soil up to this point, but this is also a nitrate leaching story. And so we know that the water loss is half of the story, because to get nitrate leaching you have to have water moving down, but the other thing is you have to have nitrate present. And we know a lot about that from our soil pit nitrate inventories. And so here we have on the x-axis the nitrate pools in kilograms per hectare, and the y-axis here is depth. And so the black line are soil pits dug immediately following harvest or at the beginning of fallow. The crop had used up most of the nitrate. There's only 12 kilograms per hectare left. A year later, we dug these soil pits, and so the blue line now is a year into fallow. There's no fertilizer applied during fallow. This increase in soil nitrate up to 46 kilograms per hectare, or an accumulation of about 35 kilograms per hectare, is all due to mineralization of soil organic nitrogen. So the microbes breaking down as soil organic matter and releasing nitrate. And you can see that the increases in soil nitrate are the largest near the surface, which is as we would expect, because that's where that really dark soil is, the black soil that's got a lot of organic matter. So the most potential for those microbes to increase soil nitrate there. So this increase of 35 kilograms per hectare, Andrew John and Clayton Jones found even larger numbers, but this accumulation of nitrate in soil from mineralization of soil organic nitrogen, very important. And we see a similar story in our lysimeter nitrate concentrations, where we have the highest concentration in that crop year following fallow, or the winter wheat year. And if we think about why this might be, we have these two general sources of nitrate to soil. There's fertilization and there's mineralization of soil organic matter. And in that crop year, we fertilize in a crop year, so fertilizer input is relatively high, but mineralization is relatively low. The crop is present, it's drying out the soil, so the microbes don't have the water they want to be happy and mineralize. In the fallow year, in contrast, we don't fertilize in a fallow year, so fertilizer is 0, but mineralization is high because the soils get wet without a crop present, and those microbes get happy and mineralize soil organic nitrogen. In the winter wheat year, or the crop year following fallow, we have high fertilization relatively because we fertilize the crop, and we also have high mineralization because we have the moist soils for that entire preceding year mineralizing soil organic nitrogen, which is carried over into the winter wheat year, hasn't had a chance to be used by the crop. So we think that that's why we see these highest soil nitrate concentrations in our lysimeters in that winter wheat year, crop following fallow. So this is soil nitrate concentrations. The way that we get to leaching rates from that is to multiply those deep percolation or drainage losses from the model that we just looked at by these concentrations, and that gives us leaching. So this is a whole additional part of the story, but the patterns are relatively similar to what we talked about for water loss. Biggest losses out of thin soils and in the fallow year, so I'm not going to go into the details on that now. There are a few interesting differences that we can talk about a different time. So the summary for paper 2 is that we have these relatively intense periods of precipitation in the wet months in the spring or during big storm events that we really get this intense loss of water through the soils, or these hot moments of deep percolation and nitrate leaching loss. We also have soils which have the thin soils that have relatively low water storage capacity, which lose a lot of water and nitrate out of them. And those that are called, we could think of those as hot spots of deep percolation and nitrate leaching loss. We also then have fallow that's overprinted on this, and it expands both hot moments and hot spots. It increases the amount of time of loss, and it increases the area of loss. And so it's these times and places of high water and nitrogen loss that dominate the landscape scale behavior. So this is a summary of what's happening for water loss from water and nitrogen loss from soils. But let's zoom back up for a minute to the global scale and think about the fact that as humans we have increased the reactive nitrogen input to soils over the last 150 years by an order of magnitude. And the only way that that reactive nitrogen gets back into the non-reactive atmospheric pool is by denitrification, which happens both from soils and also from groundwater and streams. So this is a microbially mediated process where microbes are trying to get energy, which they most commonly get from dissolved organic carbon, which is basically an electron that they're after for the energy, but in order to get that, they need an electron acceptor, which is most commonly found as oxygen. And so they undergo respiration and they produce CO2, much like we are as we're sitting here breathing. But for microbes, they can end up in an environment where dissolved oxygen gets drawn down in soil or in water, and then they're going to go to the next most energy advantageous reaction pathway or redox reaction, and if nitrate is present, they're going to use nitrate as the electron acceptor, and they're going to conduct denitrification, which releases back to the atmosphere and gives them the energy. And so, in summary, to get denitrification, microbes routing that nitrate back to the atmosphere, we need low dissolved oxygen and available dissolved organic carbon. So in the work that we've done in the study area, we have these unexplained losses of reactive nitrogen or nitrate from both soils and the riparian corridor. And that leads, and we know something about this at the global scale from work by Baumann et al., where these inputs to soil are seeing big losses back out directly to denitrification. And then also losses to denitrification from groundwater and riparian areas. And this leads us to ask in the third paper for my dissertation, what is the role of denitrification in controlling this reactive nitrogen or nitrate loss from soils, groundwater, and streams across this study area? And in order to address these questions, we're going to use an isotope data set. So just to get us on the same page about that briefly, we can think about the periodic table, which has elements defined by their number of protons. And so nitrogen is 7th in the periodic table. It has 7 protons, and 99+% of the nitrogen on the planet also has 7 neutrons, but a small fraction of the nitrogen on the planet has an extra neutron. And so this means that it's still got 7 protons, it's still nitrogen, it forms the same molecules, but it's got an extra neutron, so it's a little bit heavier, so it reacts or undergoes chemical reactions at a slightly different rate. So these are very small numbers, and we talk about them in delta notation. For example, delta N15 is the ratio of the heavy isotope to the lighter isotope in our sample versus that in a standard. And the atmosphere is our largest well-mixed pool on the planet, and so we use that as the standard. And so by definition, our delta N15 value for the atmosphere is 0. And if we have a positive delta value for nitrogen, that means that there is a higher relative abundance of the heavy isotope versus the light in your sample compared to the atmosphere. Whereas a negative number for delta N15 means that you have less abundance of the heavy isotope relative to the atmosphere. And for shorthand, I will call that light. Whereas the positive number with more heavy isotope is called heavy for shorthand. So the nitrate molecule also includes an oxygen, and so oxygen is right next to nitrogen on the periodic table. Again, 99+% of oxygen on the planet has 8 protons. All oxygen has 8 protons, 99+% has 8 neutrons, but a small fraction has 2 additional neutrons. Again, still oxygen, but it's a little bit heavier, so it undergoes reactions at a different rate, or isotopic fractionation occurs. And so what is isotopic fractionation? Well, if we think of an example that's important for our study area, if we've got nitrate in water and you have a microbe present ready to denitrify, and you've got nitrate in water flowing past it, and denitrification is occurring, we can ask, but it's not getting all of the nitrate. Some nitrate is making it past. And if we ask that microbe through a culture experiment or otherwise, it will tell us that it prefers the light isotope, which means that dinitrogen gas that it's creating through denitrification is light. in nitrogen relative to the incoming nitrate. And similarly, that nitrate that it doesn't use is heavy relative to the incoming nitrate, meaning, or in other words, these are fractionating pathways. So the products have a different isotopic composition than the incoming nitrate. And so these loss pathways, this fractionation, lets us think about some denitrification signals, isotopic denitrification signals. And the first is in dual isotopes in nitrate, where on the y-axis we have delta O18 in nitrate, and on the x-axis we have delta N15. And if denitrification is happening, we expect a positive correlation between these two variables. And that's because as a microbe is denitrifying, it's discriminating against the heavy isotope, and so what gets the nitrate left behind is getting heavier. It's discriminating against nitrate molecules that are heavy with both respect to nitrogen and oxygen. And so the nitrate molecules left behind are relatively heavy, and they fall along this line with positive correlation in dual isotope space. Our second signal has delta N15 on the y-axis and nitrate concentration on the x, and we expect a negative correlation here, where in a classic closed system, you've got a bottle of water with nitrate in it. The microbes start to use the nitrate for energy, so they reduce the concentration, and they prefer the light, so they're leaving the heavy nitrate behind, and you get less and heavier nitrate present at the end. So these are our two signals. If we plot our data starting with soils, all of our soils data show a shotgun pattern, no signal of denitrification present. But if we focus in on these black crosses, which are the lysimeter samples from deep installations during the fallow year, we see this signal of denitrification. Why might that be? Well, if we think about the fallow year, it's relatively wet compared to the crop year, and soil moisture, no crop present, so the soils get wet. Soil moisture is a primary control on dissolved oxygen in soil. Wet soils restrict the ability of atmospheric oxygen to move into soils. So it makes sense that we see this denitrification signal during fallow periods. If we look at our second signal, sure enough, we see the negative correlation or a denitrification signal, a strong line of evidence with both of these signals present during fallow. If we add groundwater on now, specifically our groundwater with high dissolved oxygen, we see no denitrification signal. What we do see is a clustering of those right in the center of our soil pattern. And this is a beautiful illustration of the fact that this is an isolated landform where groundwater quality is determined by the overlying soil chemistry that's where groundwater recharge is happening through those soils. And so similarly for our second plot over here, we have a mean nitrate concentration of 20 milligrams per liter and a delta N15 around 4 and no signal of denitrification. Whereas if we add on all of our groundwater samples where we have some with very low dissolved oxygen, we see a strong denitrification signal, this strong positive correlation, and a strong negative correlation here for those low DO samples. So while in the majority of our groundwater in the study area we see no denitrification because there's high oxygen, in these few riparian groundwater sites where dissolved oxygen gets drawn down, denitrification is going almost all the way to completion. It's getting almost as low as 0, and this strong N15 signal is suggesting that is denitrification loss. So if we now add on our stream data, we see in the darkest green here is Louse Creek. We see a strong denitrification signal in both of our diagnostics here. Porter Creek, the lighter green, we see a subtle denitrification signal in that the symbols are moved slightly up and to the right from the groundwater origin, but a much lower signal in Porter Creek than in Louse Creek. An interesting nuance that I don't have time to get into just now. But so all of this nitrate in this system is being lost from soils. And so let's return to soils for a moment, where we said that we have this signal of nitrate lost from soils to denitrification, but we've also talked about how we have this gain of nitrate, or this accumulation over the fallow year of 35 kilograms per hectare. And so this pattern we see actually in our lysimeter samples as well, where early in the fallow year we start at low nitrate concentrations, and then it's late in the fallow year that we have the higher concentrations. So evidence of this nitrate accumulation in our lysimeter samples as well. So signals of both losses and gains. And if we were only looking at the nitrate inventory in the soil and weren't looking at the isotopes, we wouldn't know that this loss was happening. So it's the isotopes that are telling us that this denitrification loss is happening. So let's think just generally about this range of 0 to 10, delta N15 in nitrate in soil, and think about what this can tell us about losses. So in soil, we've observed this range of soil nitrate, 0 to 10 per mil. And we also know from observations that soil organic nitrogen is around +5. And during a fallow year, the only way that nitrate gets into soil is by the process of decomposition of soil organic nitrogen. We can ask these microbes, they'll tell us that they prefer the light soil organic nitrogen. And the degree to which they prefer the light, which is a general light preference, is through this epsilon value, which we'll take an intermediate value for now of -10, which we can simply add to the value for soil organic nitrogen, and that tells us we would expect a -5 value coming into soil. We never see values that low. We never see values below 0, suggesting that there is a concurrent loss, a fractionating loss that is leaving behind heavy nitrate. And so again, if we have these denitrifiers that prefer the light nitrate, they leave the heavy stuff behind. This is a fairly strong preference for the light isotope. We'll take an intermediate value here. And so it's my hypothesis that the reason we're not seeing these lighter values in soil is because the soil has undergone denitrification, and that's what's leaving the heavy stuff behind, so that we're never getting those lowest values. So if we accept these assumptions about the fractionation factors, we can combine that with our 35 kilograms of accumulation to estimate that there are 10 to 50 kilograms of nitrogen per hectare per year. There's enough uncertainty in these assumptions that this is basically a back of the envelope calculation currently, but these losses are large. They're up to more than two times the leaching loss rates, and are up to more than half of fertilizer application rates. And so the implications here are very important to nitrogen losses from this system, and they're poorly understood. In summary, for this third paper, we see these strong signals of denitrification loss in riparian areas, which are beneficial to water quality. They're reducing the nitrate loads, but these are deleterious to climate, because certainly some of that denitrification is not making it all the way to N2, but is stopping at nitrous oxide, which is a strong greenhouse gas with a long residence time in the atmosphere. In soils, we have more nuanced signals, but estimated rate, back of the envelope calculations is 10 to 50 kilograms per hectare, which are large loss rates that are deleterious to soil fertility, a lost opportunity for crop production, and again are deleterious to climate. These soil losses are masked during fallow by net accumulation of nitrate. So we have these big losses, but you can't see them if you're just looking at the accumulation of nitrate. This denitrification loss from soils is certainly important to the mass balance in our system, and I would argue globally, because of the 10% of the Earth's terrestrial surface that is cultivated, that area spends 50% of the time in fallow. So overall conclusions, at the landscape scale, we've estimated losses of 10 to 20 kilograms per hectare, which are at important rates relative to fertilizer application rates. But again, this is also derived from soil organic nitrogen. In soils, these thinnest soils are hot spots for nitrate leaching, and fallow exacerbates those losses, where in these thinnest soils, they're storing no water on average during a fallow year, and so are good candidates for either reduction of fallow or even conversion to perennial vegetation. Denitrification losses in riparian corridors is highly variable, and in some places is up to 100%, suggesting opportunities to leverage riparian management for water quality benefits. But these losses in the riparian corridor are small relative to the more spatially expansive losses from upland soils. And those losses from upland soils, both during fallow and otherwise, are poorly understood and could be very important losses that we need to understand better. This has been a research talk, and there are a lot of opportunities for further research that come out of this. One is the role of macropores in understanding water and nitrogen loss from soils. Definitely important and a useful topic for future research. I think it's interesting to think about the thickness of soils and the connection to economic returns, where for some of those thinnest soils, you might not ever get enough yield profits back from your yields to outweigh the inputs of both seed and nitrogen fertilizers. So interesting to think about those thresholds for where the thinnest soils don't make sense for inputs. Those differences in denitrification losses between Louse and Porter Creek are very interesting. We have a lot of ideas about that would be interesting for further study. And denitrification during fallow and even more broadly in soils is poorly understood and could be very important to losses. And the EPSCoR Cruise project is a new project that Stephanie Ewing is leading an agricultural component of in the Judith area, doing some really interesting interdisciplinary research, building on some of these findings. I'm really excited about some future outreach work, just getting back into the watershed and talking to our cooperative farmers and others about some of these newest results is exciting to think about. The town of Denton has some nitrate in their drinking water, and there's already been a few conversations about how some of our results might be insightful to try and help address some of their issues. The soil modeling work that have been applied here could readily be applied to many other areas and any other types of questions. And I think it's fun to think about possibly an interactive web tool where producers could select their soils, their rotation, and a weather station and see some plots similar to what we've been looking at or catered more specifically to what they're interested in. And with that, I have a lot of acknowledgments to make. I've already mentioned my committee and the funding sources. Co-authors here, critically important to the success of this work. The Ewing Lab Group, Christine Miller's thesis work was foundational for a lot of the work that I presented here today, and the camaraderie and support of these folks in the lab has been incredible. The participatory research team that I mentioned, this work would not have been possible without them. Jane and Christine, the folks in the Environmental Analytical Lab, did the analysis. The graphics, Marco responsible for the landscape graphic that I used today, and field technicians, incredible support from a number of field technicians. Kyle Marins was pivotal in year one of the project, and then Simon Fordyce in all of the other years of data collection was the lead and just an incredible force getting that data collected. And Jim Botter, who was at my defense, and Steve Custer. Jim was my first mentor in Montana and explained to me what fallow was, and incredible insights from Dave Wichman and Steve Custer. And my family, my parents were at my defense, and I like to recognize them, as well as my partner Brooke, who made the refreshments, my friends, and I can't imagine having made it through this process without my therapist, and the yoga team at Bend Beyond helping keep me sane. And with that, I'd love to take any questions. And then, why not? Gotta get in there, look around if you're gonna find the answers to these questions. What? Thank you for listening.